Broadly speaking, my research program tries to understand the molecular language that cells use as they work together to build and remodel the brain and health, and then how these really powerful interactions go awry in the context of neuro-oncological diseases. And so I want to tell you one story today in two parts about the way in which neurons interact with the oligodendroglial cells that ultimately form the myelin sheath to increase the speed of neural impulse conduction and provide metabolic support to axons, how we're beginning to understand that this is a experience and activity regulated process that tunes neural circuit function throughout life is a really important mechanism of plasticity. And then how these really powerful interactions between neurons and cells of glial lineage can be hijacked in the context of malignant glioma. I'm a pediatric neuro-oncologist, and I focus on gliomas of childhood. And when we take a step back and consider the major gliomas of childhood, it becomes really clear that these tumors occur in certain parts of the nervous system at certain ages. And this really speaks to an underlying neurodevelopmental process that has gone wrong. So for example, the kind of optic pathway gliomas that occur particularly in the tumor predisposition syndrome neurofibromatosis type 1 tend to happen in very early life, in early childhood. And here I'm showing you an optic pathway glioma in a 4-year-old with NF1. One of the worst human cancers, diffuse intrinsic pontine glioma, which is an H3K27M mutant diffuse midline glioma of the brainstem, one of the leading causes of pediatric cancer-related death. This tends to occur in mid-childhood, whereas a molecularly related tumor that occurs in the thalamus, often involving both thalami diffusely, tends to occur slightly later in childhood, peaking in incidence around age 10. And then the hemispheric high-grade gliomas happen in adolescence and young adulthood, and then continue on in the same pattern throughout adulthood. And when we take a step back from this and we think about the neurodevelopmental processes that this pattern kind of fits with, it becomes really clear that this pattern of tumorigenesis in the childhood nervous system maps pretty well onto the developmental and spatiotemporal pattern of myelination, which is actually a developmental process that spans more than 30 years of neurodevelopment. So I can't see the audience, but I imagine that there are some students in the audience right now who have not yet completed developmental myelination. So this is a very, very protracted process. And this matches really, really closely the pattern with which these tumors arise. So at a time when there's a discrete wave of myelination in the ventral pons, myelination of the corticospinal tracts, around ages 6 to 8, this is when diffuse intrinsic pontine glioma occurs, and when there's active myelination of the neocortex and its projections in adolescence and young adulthood, these are when hemispheric high-grade gliomas tend to occur. And these observations are concordant with findings from my laboratory and from many others that these tumors, these glial malignancies we've traditionally thought of as astrocytomas, in fact, appear to arise frequently from precursor cells in the oligodendroglial lineage, either early oligodendrocyte precursor cells or just less committed cells that are going to go down this oligodendroglial lineage, and certainly fits with molecular observations at the single cell level that a major subpopulation of all these different forms of tumors very, very closely resemble the normal oligodendrocyte precursor cell. And so we may learn really important lessons about what normally regulates gliomagenesis by better understanding this process of gliogenesis. And so for over a decade now, my laboratory has been trying to understand what does regulate the proliferation and functional differentiation of healthy oligodendroglial cells, and then how is this subverted in the context of glial malignancies. And so there's a really intriguing idea that had been in the literature for some time, first introduced in the 1990s by Ben Barres, that axons of neurons themselves may regulate the extent to which they're myelinated, and that this may happen in an experience and activity-dependent manner. And certainly, there was some early evidence that correlated activity and experience with myelination, but it was a very controversial idea because there are also very clearly activity-independent modes of myelination. So now more than 10 years ago, we started to ask the question, does neuronal activity regulate myelination or modulate myelination in an experience-dependent way? And how do these putative neuron-glial interactions then influence the cancer? And so to answer these questions, we've leveraged techniques in modern neuroscience like in vivo optogenetics, and that allows for control of targeted populations of neurons controlled by light. And so here you see this mouse executing complex motor output, and the mouse is doing that because we have expressed a light sensitive ion channel, in this case channel rhodopsin 2, a blue light sensitive cation channel, in cortical projection neurons, and then delivered pulses of light over the motor planning area. And if we do that at the right frequency, then the mouse begins to execute complex motor behavior, and we can begin to ask questions about how other cell types within the stimulated circuit respond to changes in neuronal activity. And what we found when we looked at the healthy oligodendrocyte precursor cells is that there was a robust circuit-specific proliferation of oligodendrocyte precursor cells just within those cortico-callosal projections of the premotor circuit. And if we fate map those cells over time by administering the thymidine analog EDU at the same time as optogenetic stimulation, then we see that new oligodendrocytes are made, and that the myelinated ultrastructure of that circuit changes, and it changes in a way that we would predict may alter circuit function and influence behavior. And indeed, we found changes in motor functional behavior that depended upon this addition of new oligodendrocytes and changes in the myelinated ultrastructure. And so this plasticity of myelin, this really powerful neuron-glial interaction in the healthy brain, tunes neural circuits to promote coordinated circuit function. This is mediated in part by activity-regulated paracrine factor secretion, like brain-derived neurotrophic factor. There are also these really fascinating synapses, really electrophysiologically functional synapses that form between neurons and oligodendrocyte precursor cells all along the axon tract. The role of these in activity-dependent myelination is still somewhat enigmatic, but they exist on the majority of oligodendrocyte precursor cells. And these adaptive changes in myelin contribute to cognitive function, to attention, learning, and memory. But could this be subverted in brain cancer? Could these OPCs, if they possessed an oncogenic mutation, could the glioma cell actually be regulated by neuronal activity? And so to ask that question, we repeated that very same experimental paradigm, optogenetically stimulated cortical projection neuronal activity, but this time in the context of a diffusely infiltrating pediatric cortical blastoma isolated from a patient of mine. And what we found was that the malignant glioma cells indeed increase their rate of proliferation in response to neuronal activity, and that this results in an overall increase in tumor burden, specifically within the stimulated circuit. So brain activity can regulate brain cancer growth. We, in later years, in collaboration with David Gutman's lab, turned to a model of low-grade glioma. This is a glioblastoma. We looked at a low-grade, grade 1 pilocytic astrocytoma model that occurs in the optic nerve of children with neurofibromatosis type 1 and in a genetically engineered mouse model of neurofibromatosis type 1. And in this mouse model developed by the Gutman lab, there is emergence of these tumors very reliably at 9 weeks in the vast majority of mice, and about 95% of mice. And so what we did was we optogenetically stimulated optic nerve activity just prior to the onset of these tumors. And what we found is that the mice that had elevated optic nerve activity through this optogenetic paradigm developed much larger tumors than their littermate controls that we identically manipulated, placed the optical fiber, just never turned on the light. Now, this optic pathway glioma model is a really exciting opportunity to ask questions not only about tumor growth, but also about tumor initiation, because we know exactly when and where these tumors are going to form. And actually, they're in an incredibly tractable neural circuit in the optic nerve, which is composed of one type of axon, just retinal ganglion cell axons. We can actually modulate optic nerve activity using visual experience. We don't actually need to use optogenetics. There are natural opposites in the eye, and we can just modulate the activity of the optic nerve with light exposure. ambient light exposure. And so what we did is we took these mice and we placed them after the optic system had developed at around the time of, that the tumors develop at 9 weeks of age in complete darkness. So rather than living in 12 hours of light, 12 hours of dark, we put them in 24 hours of darkness. And what we found that if we did that at 9 weeks or even just after tumor initiation at 12 weeks, then the tumors that formed were far fewer and much smaller than their littermate controls raised with normal visual experience. The way that I'm showing you the tumors here is to plot the volume of the optic nerve on the x-axis by the proliferation index in the optic nerve. And these low-grade gliomas tend to, on this graph, cluster here in the right-hand quadrant. The gray hatch mark shows you the volume, optic nerve volume and proliferation index in wild type optic nerve. So anything that falls within this gray, we would assess as not actually having a tumor. So we then wondered, what if we put these mice in darkness to decrease their visual pathway activity prior to the onset of these tumors? Might we actually avoid them entirely? And indeed, if we placed mice in 12 hours of, from 12 hours of light and 12 hours of dark into 24 hours of darkness at 6 weeks of age, then no tumors formed, even if after what appears to be a susceptibility period, we replaced mice in normal visual experience conditions in 12 hours of light, 12 hours of darkness, really underscoring how powerful these interactions are, that in the context of this oncogenic mutation that so reliably produces an optic pathway glioma in this mouse model, that just modulating the activity of the optic nerve is sufficient to prevent tumor initiation and maintenance. So what are the mechanisms that are mediating these neuron-glioma interactions, either in high or low-grade glioma? To answer that question, we performed a really simple experiment. We took explants, either the healthy mouse cortex or the healthy mouse optic nerve with retina, and we placed those explants into simple medium, and then collected the conditioned medium when the neurons in those explants were at varying levels of neuronal activity, either optogenetically stimulated, spontaneously active, or silenced with tetrodotoxin. And what we found was that when we placed that conditioned medium containing secreted factors, and we placed that on cultures of glioma cells, that there was an activity dose-dependent increase in glioma proliferation abrogated when neuronal activity was silenced with tetrodotoxin. And that this effect of neuronal activity regulated secreted factors was conserved across multiple different forms of high and low-grade glioma. Summarizing, a couple years of work to identify what it was in that conditioned medium that was mediating this proliferative effect, we found that there were two main factors. Not unexpectedly, because it plays such a critical role in healthy neuron-glial interactions, we found that brain-derived neurotrophic factor serves as a mitogen for glioma cells. This was not a surprise, but really surprisingly, we found a protein in a shed form called neuroligin-3. Now, neuroligin-3 is a well-known postsynaptic adhesion molecule, but it wasn't known to be a mitogen in any context, and actually it wasn't even known to be secreted. And so we discovered that neuroligin-3 is being cleaved by activity-regulated ADAM10, this metalloprotease, at the membrane, resulting in release of this N-terminal ectodomain that then binds to the glioma cancer cell. But how important is this mechanism? There are many cell intrinsic as well as microenvironmental mechanisms that regulate tumor progression. And so we wanted to test the relative importance of this microenvironmentally released neuroligin-3. And to do that, we xenografted brain cancer cells into the environment of either the neuroligin-3 wild type or neuroligin-3 knockout brain. And really unexpectedly, what we found was that in the absence of neuroligin-3 from the tumor microenvironment, that there was simply no progression of these xenografted cells, and that this apparent dependency on neuroligin-3 in the tumor microenvironment was conserved across multiple different forms of high-grade glioma, including pediatric glioblastoma, diffuse intrinsic pontine glioma xenografted either to the pons or to the cortex, and adult IDH wild-type glioblastoma. We also found that this dependency on neuroligin 3 was operant in the optic nerve and was really necessary for the progression and initiation of optic pathway gliomas. What we did find, though, was that this dependency did not extend to patient-derived models of breast cancer brain metastasis, that if we put patient-derived models of breast cancer brain mets into the mouse brain lacking neuroligin 3, that they grew just fine. So why is this such an important mechanism? Well, we know that when neuroligin 3 is cleaved in this activity-regulated mechanism and binds to the glioma cell, that it recruits numerous oncogenic signaling pathways. There's early upstream stimulation of focal adhesion kinase, downstream SARC-RAS, and PI3 kinase pathways. But that explains the sufficiency, not the necessity of neuroligin 3. So digging deeper, what we found was that there was regulation of a number of synapse-associated genes. There's a feed-forward effect of neuroligin 3 on its own expression, together with upregulation of the BDNF receptor TrkB, but also a number of different glutamate receptor subunits and other kinds of synapse-associated structural proteins. And so this raised for us what seemed at the time kind of a crazy idea that, like there are synapses between neurons and healthy glial cells, there might also be synapses between neurons and malignant glioma cells. And indeed, we see these synaptic structures when we look by immunoelectron microscopy, in which we can unambiguously identify the malignant cell. We see these very clear synaptic structures. Testing the idea that neuroligin 3 may be functioning to regulate these, we find that far fewer of these neuron to glioma synaptic structures form in the absence of neuroligin 3 from the tumor microenvironment. But are these synapses actually functional, or are they simply a shadow of the cell type from which we believe these tumors emerge? To test this, we performed whole cell patch clamp electrophysiology of the tumor cells xenografted into a very tractable and manipulatable circuit in the hippocampus. And what we found is that if we patch clamp recorded from these tumor cells xenografted to the CA1 region of the hippocampus while electrically stimulating the axonal afferents into CA1, that there were very clear synaptic currents. These are morphologically, excitatory postsynaptic currents. They depend upon action potentials. They're blocked by tetrodotoxin. They exhibit multiple electrophysiological characteristics of bona fide synapses, including paired pulse facilitation, quantal single vesicle events called mini EPSCs, and more specifically, this very first kind of neuron-glioma synapse that we identified is a calcium permeable AMPA receptor dependent synapse. At the same time that we discovered these in pediatric diffuse intrinsic pontine glioma and pediatric glioblastoma, Frank Winkler and Thomas Kunar's group in Heidelberg discovered the same thing in adult IDH wild type glioblastoma. And so we're finding that these cancers are not just driven by neuronal activity, they're actually integrating into neural circuits. They are doing this both through bona fide neuron to glioma synapses, as well as electrically through activity-dependent potassium evoked currents that are then amplified through a tumor-to-tumor gap junction coupled network. And I want to point out that the neuron to glioma synaptic integration is a dynamic process that's modulated further by activity. And we've found that these synapses are both elaborated and reinforced through mechanisms much like long-term potentiation modulated by malignant synaptic plasticity. So there are multiple ways by which these tumor cells are undergoing membrane depolarization. And so we hypothesize that it's actually the voltage change itself. It's the membrane depolarization that may really matter to the tumor cell. And that would make sense because we know that there are voltage-dependent mechanisms regulating healthy neural stem and precursor cell proliferation and growth. And so we turned again to optogenetics, optogenetically stimulating not the neurons in the microenvironment, but this time expressing those light-sensitive ion channels in the tumor cells, and then optogenetically depolarizing the tumor xenograft. And indeed, just membrane depolarization increases the proliferation rate of these tumors, while conversely blocking neuron to glioma synaptic communication, either pharmacologically or here genetically, has a robust growth inhibitory effect. We can visualize this electrical activity in the tumor using genetically encoded calcium indicators. And I think this particular calcium imaging showing you the electrical currents in the cancer cells using calcium transients as a surrogate really underscores this new understanding that these cancers are actually electrically active tissues. And that is not how we have been approaching them. We now, as a field, need to understand the mechanisms of malignant circuit assembly, plasticity, and evolution over the disease course. To uncover the granular details about the voltage sensitive mechanisms of tumor cell proliferation. And I propose that this will give us important insights for brain cancer therapy. Already, as we consider what we know, a number of targets are beginning to emerge for therapy, including neuroligin-3, AMPA receptor-dependent synaptic signaling, gap junctions, neurotrophin signaling. I didn't mention it, but also GABAergic signaling in the context of certain malignancies, potassium channels, and synaptogenic factors. I want to end with the idea that the interactions between neurons and brain cancer cells is a bidirectional one. Just as neuronal activity drives glioma growth and proliferation through both paracrine and through synaptic and electrical mechanisms, the gliomas increase neuronal excitability and activity, so creating this kind of malignant cycle of neuronal activity dependent glioma growth and glioma dependent neural remodeling. I think that the mechanistic parallels that are evident between normal neuron-glial and neuron-glioma interactions really underscores the extent to which these tumors are hijacking mechanisms of normal neural development and plasticity, and demand that we begin to approach these cancers from a neuroscience perspective. And I'll end with the idea that the neuroscience of cancer is critically important, not just for cancers of the central nervous system, but increasingly we're understanding that these interactions are at play in cancers very broadly throughout the body, through the peripheral nervous system, and through systemic neural signaling to the cancer. And there's a number of different cancer types that have now been demonstrated by a range of groups to be really potently modulated by the nervous system. And so emerging then is the concept that the neuroscience of the cancer really matters and is a pillar of what I think we will need to target for effective therapy, particularly of very aggressive cancers. I want to just quickly point out the people who did this work in my laboratory, some of whom have now moved on and started their own independent laboratories, Hamza Venkatesh and Yuan Pan, both fantastic scientists in their own independent labs. If anybody out there is looking for a postdoctoral fellowship, I highly encourage you to reach out to them. Katie Taylor and Tara Barron currently in my lab. Others and our collaborators, funding sources, and of course the patients and families whose donation of tumor tissue enables all of the work that we do. Thank you so much for your attention, and hopefully we have some time left for questions.